This is a presentation on proteins for cellular biology. This assignment is supposed to be the equivalent of a 5-minute verbal presentation, so I'm going to be mainly hitting on the high points and things I think will be relevant for testing. Starting with amino acids, proteins are made of amino acids. Another way to say this is that amino acids are the monomers to proteins. Amino acids have a central carbon, which is attached to an amino group, a carboxyl group, a hydrogen, and a functional group. There are 20 different amino acids, and each one has a unique functional group, which determines how it interacts with its surroundings and the intermolecular forces. For the formation of proteins, amino acids are linked with peptide bonds, linking the carboxyl group of one amino acid to the amino group of another amino acid, and this is done via a condensation reaction. Proteins fold into 3D shapes based on the specific order of amino acids. This is due to intermolecular forces, which creates particular structural motifs. The four interactions that drive this folding are hydrogen bonds, which is a bond between a hydrogen connected to a particularly electronegative atom like oxygen, which has at least one free set of lone pair. Electrostatic attractions, which can occur between any two polar substances. Van der Waals forces, referred to commonly in chemistry as dispersion, which are induced dipoles based on the random movement of electrons. To get a better understanding of this, looking into how geckos' feet work gives a pretty clear visual image. And hydrophobic interactions. Some of the amino acids have nonpolar functional groups, and these nonpolar functional groups don't tend to interact with water as much because they can only interact through dispersion, and water is particularly strong because of its hydrogen bonding. So they tend to clump in the middle and avoid water, while the polar functional groups are around the outside where they can interact with water. So in proteins, the order of amino acids impacts the shape of the protein, and the shape of a protein impacts its function. For some examples of types of proteins, we've got receptors, which mediate signal between the inside and outside of a cell, allowing it to interact with its surroundings, receive and interpret signals. There are transporters like hemoglobin, pictured on the right. Those move around particular substances to the places they need to go. Hemoglobin specifically moves oxygen. And there are motor proteins which mediate movement of cells in larger structures. There are also enzymes like that RNase pictured on the right. RNases are a type of enzyme that break down RNA. And there are pumps and other specific proteins in the membrane, like the sodium-potassium pump, which move things, allow things to move in the direction they need to go. The sodium-potassium pump particularly moves against the concentration gradient to restore the balance of the cations that are interacting with it. So for protein structures, we've got primary structures, which is the order of amino acids, so just the sequence that it takes place that it's in. Secondary structures, which are particular shape features and structural motifs like beta sheets or alpha helices. Tertiary structures, that refers to the 3D shape of a single protein or its conformation. And quaternary structures, which refers to a structure made up of multiple peptide chains forming into a protein complex. So making a protein, every 3 base pairs of DNA are a codon. Each codon represents start, stop, or amino acid. RNA polymerase transcribes the base pair sequence from DNA from a start to stop codon, forming messenger RNA. This RNA goes to the ribosomes, which then match up each codon with the proper amino acid. Of course, the process does have more complex facets than this, and this is just a brief summary in order to maintain brevity while still getting the concept. And it doesn't really go into the whole concept of promoters and transcription factors, which we'll likely get into later in the course. And I've included a codon chart there, just so that you can visualize the different order of codons resulting, the different order of base pairs resulting in the formation of different amino acids. All right, that is all for this presentation. Thank you for listening.